Defense against infectious diseases. Keep pathogens out. The basics. Often we previously develop immunity to any pathogens. Antibodies work against living bacterial cells and do not affect our bodies. Any living organism or virus is capable of causing diseases. It's called a pathogen. Exposure to pathogens does not always result in disease. Humans are already well defended from pathogens. The skin membrane. Pathogens can enter through the skin. The epidermal layer, mainly of dead cells, form a good barrier against most pathogens. As long as the skin remains intact, we are protected from most pathogens. This is why it's necessary to clean and cover cuts of the skin. The mucous membrane. Pathogens can enter the body through the lined tissue cells that form a mucous membrane. Mucus can trap incoming pathogens and prevent them from reaching the cell. The cilia lining movement traps pathogens up and out of the mucus-lined tissues.
Blood clotting. It minimizes the chances of infection and blood loss. When blood vessels are broken in the skin, pathogens have an entry into the body. Body's response is to clot and seal the damaged blood cells to prevent excessive blood loss and prevent pathogens from entering. Plasma proteins within the blood have two clotting properties, prothrombin and fibrogen. They remain inactive until needed. Platelets, also within the blood, with the red blood cells and white blood cells, come to the open passage to clot. When a small open vessel is damaged, the damaged cells release the chemicals that stimulate platelets to adhere to the area, which release chemical clotting factors that convert prothrombin to thrombin. Here's an illustration. Damaged blood vessels release the chemicals that allow the platelets to adhere to damaged areas, and then the chemicals convert and from prothrombin to thrombin and then from then the fibrogen goes to fibrin, forming a mesh. The mesh doesn't allow for more bacteria to come in, and it also stabilizes as more platelets come in. When pathogens get past skin and mucous membranes, when they first enter, the first response is known as the primary immune response. It usually takes a week or more for it to successfully eliminate the pathogen, and this is usually when symptoms emerge. Secondary or third encounter with immune system is quick and more intense, so symptoms aren't really experienced. Role of phagocytic white blood cells. In the bloodstream, white blood cells help fight off pathogens that enter more than once. Macrophage is a large white blood cell that are able to change their shape and surround an invading cell. Here is an illustration of how it's done. The helper T cell, specific to the antigen, is activated by the macrophage. As you can see, it's going around the pathogen and circling it, and as it moves throughout, the B cells divide repetitively to produce antibodies, secreting plasma cells. Antibodies produced by the clone of the plasma are specific to antigens on the pathogen and help to destroy it. B cells also divide to produce memory cells. The memory cells are part of primary and third response, so if the pathogen comes back again, it won't take as long to fight it off.
Vaccination. Active immunity can be acquired artificially through vaccination. The vaccination containing a live or weakened version of the pathogen or derivative of it, stimulating a primary immune response. In the future, if the same one revisits the body, the antibodies destroy it in a secondary immune response, which can also be done by a booster shot. Antibodies produced by lymphocytes. Immunity. Antibodies are protein molecules that are produced by the body in response to a pathogen. Each antibody is different because the pathogens are different. Not self-proteins are antigens. They trigger an immune response. The antigens are Y-shaped with a binding site on the end of the forks. This is where the antibody attaches itself to an antigen and pathogen. Our cells produce a relatively small number of antibodies in comparison with the massive infection that may be in the body.
What is HIV and effects of the immune system? HIV stands for Human Immune Deficiency Virus. It's specific about which organisms and which cells it affects. The host cells are usually humans, and it attacks the lymphocyte cell types involved in the immune system. An infected person has a severe drop in the lymphocyte population, losing ability to produce enough antibodies, leading to AIDS. After a few years of having AIDS, AIDS begins to infect the person, so no longer they can fight off pathogens. There's no cure for HIV, but some are available for the infections that AIDS cause. HIV is transmitted person to person by sexual intercourse, during which the mucous membranes go from one person to another, transfusion of infected blood, and sharing of needles. Social implications of AIDS are severe. Sufferers are stigmatized and family can lose members. Parents can be lost and grandparents take care of the kids or orphaned, leading to economic productivity is being lost. Treatment costs are so high in certain countries that are poor and have high infected population, causing the disease to spread. Here you can see the HIV particles on the T cell, which is the lymphocyte cell.
Okay. Antibiotic combat to bacterial infections. Antibiotics are chemicals that take advantage of the difference between prokaryote and eukaryote cells. Some block the bacteria's ability to grow and divide. It has no effect on human or animal cells. Some selectively block the biochemistry needed by the bacteria, and some selectively block protein synthesis in bacteria.
Bacterial resistance. Bacteria shows genetic variations just like all other living things. A few variant can then reproduce and repopulate a colony in a very short period of time with bacteria that are all resistant to the antibiotic. The resistant ones create a new strain of bacteria. This happens from long-term use and overuse of antibiotics.
This is the end of Defense Against Infectious Diseases.
Thank you for watching. Bye.